And then we'll talk about how you would have done things in lab. Because this week, we would have started some really fun stuff. So we would have started a unit on normal flora, and you guys would have been the guinea pigs, and we would have tested your throat, see what's growing in your throat. We would have tested your skin to identify which species of Staphylococcus you have growing there, and your teeth and gums, right, to see your relative risk of cavities. So we would have done all that fun stuff starting this week, but instead we'll just talk about it. So we'll start off with throat cultures. And for the throat, we use a medium called blood agar. And if you refer back to the table that summarizes selective and differential media, you'll see that's where blood agar is listed. And blood agar is basically just ordinary TSA, tryptase soy agar, plus 5% sheep's blood. And so the blood enriches the agar. Because blood has a lot of nutrients in it, right? Proteins, carbohydrates, fats, purines, pyrimidines, all kinds of growth factors. So there's lots of good stuff in blood. And so if you have an organism that needs additional nutrients, it may not grow well or grow at all on plain TSA. But you add blood to it, and then those microorganisms will grow. We call those microorganisms that have particular growth requirements fastidious. So a fastidious bacterium may not grow on plain TSA, but you add blood to it, and then the organism will grow. So the blood enriches the agar to help fastidious organisms grow. Now, the other thing about blood is that we can determine the type of hemolysis or hemolysis, right, that occurs. Remember when we talked about disease and pathogenicity, we talked about how some pathogens produce hemolysins, right, those toxins which can destroy red blood cells. So as it turns out, there are actually many different bacteria that can destroy red blood cells, at least to some degree. And so we can determine the type of hemolysis by looking on the blood agar plate. And you see in these images, we have three types. If you're familiar with your Greek alphabet, this first symbol here is alpha, and then beta, and then gamma. So those are the three types of hemolysis. Alpha hemolysis is the partial breakdown of the hemoglobin, and that partial breakdown turns the blood kind of a greenish brownish color. And so we can see that more easily here on this middle plate, right? The greenish brownish color, to the agar. But that's the partial breakdown of the hemoglobin. Beta hemolysis is the complete destruction of the red blood cells. In fact, the destruction is so complete, you can actually see through the agar plate. So here you see how this beta is nice and bright, a nice bright halo all around it because all the blood's been destroyed. In this plate here, I can actually see her finger behind the plate now, right, because you can see through the agar. So beta hemolysis is the complete destruction of the red blood cells. And then gamma hemolysis is actually no hemolysis at all, but scientists like to give it a name, and so they call no hemolysis gamma hemolysis. And so the blood agar is unchanged. It's still nice and red underneath and around the bacterial.
Yeah, Carter. Why don't you find Grant?
Somebody with their microphone on. Right, so remember, alpha is the partial destruction, beta is the total destruction, and gamma is no destruction at all. So what kinds of bacteria produce these results? Well, when we do this in class, what we find is that all students have alpha hemolysis, so everyone has alpha. A few students will also have beta, and that normally concerns them a lot when they see that they have bacteria in their throat, which are completely destroying their red blood cells, right? But as it turns out, there are common ordinary things like Staph aureus. Staph aureus is beta hemolytic. And if you like to eat with your fingers and hands, then you may have some Staph aureus in your oral cavity. So that's how you got it on your blood agar plate. Bacillus cereus, an organism found in the environment, also one that we tend to eat because it can be, we got spores on our food, on our hands. Bacillus cereus in particular likes rice. So it's not unusual, for there to be bacillus in someone's throat. And the bacillus colonies would normally be relatively large colonies. You can easily recognize them. The Staph aureus is kind of an intermediate sized colony. And then if there were really small, bright white colonies, that's Streptococcus. But it could very well be an organism, Streptococcus mutans, which is found on people's teeth. It's actually the most common cause of cavities is Streptococcus mutans. So it doesn't mean you have a nasty form of strep like strep pyogenes, but maybe just Streptococcus mutans from your teeth. So it's not unusual to find beta hemolysis in throat cultures. And it can come from, fairly common organisms that don't normally cause any problems. Now, sometimes students get weird things on their blood agar plates. And if we were doing the lab after you got your results back, if you had something weird and you wanted to do a gram stain on it, you could actually stain it and take a look and see what it is. Because one semester there was a student had these really mucoid colonies, really slimy, colonies. And when the student did the gram stain, the most enormous rods I have ever seen. I have no idea what that organism was. Never saw it before. And then another student, her plate had a definite fruity smell. So it smelled kind of like fruit, which is actually pleasant, except that it's a bad microorganism that causes the fruity smell. And it's a pathogen, Pseudomonas aeruginosa. If you remember from the growth patterns experiment, the green extracellular pigment was produced by Pseudomonas aeruginosa. And I had to go look that up when I smelled her plate because I would not have imagined Pseudomonas would be in anyone's throat. And it is rare. Only about 5% of people can you find Pseudomonas aeruginosa. So you never know what you're going to find in a throat culture. So that's what makes it interesting for us to do on students so they could see, what they have growing there. But the main idea is that the blood enriches the agar, and that allows fastidious microorganisms to grow. We can also differentiate the type of hemolysis. Alpha hemolysis, the partial breakdown, beta, the total breakdown, and gamma, no breakdown, no change in the blood at all. So any questions on blood agar plates?
Professor, is it after the blood is enriched that we'll be able to see the hemoglobin in it, or it doesn't matter?
Well, the agar is enriched with the blood. All right, so the blood enriches the agar to provide extra nutrients, and that allows fastidious microbes, those that are harder to grow, to grow on the blood agar plate. And in addition, if the bacteria eat blood, we can see that. We can differentiate the type of hemolysis. So does that answer your question, Christiana?
Yes, thank you.
Any other questions about blood agar?
Did any students ever have strep throat in class?
Well, we can't tell, right? Because all we know is that if it's a small white colony that's beta hemolytic, it most likely is some type of streptococcus. And when you do a gram stain, you do see beautiful long chains of cocci. But whether or not it's strep pyogenes or just strep mutans, we can't tell. So there's no way to know for sure.
Oh, okay.
You'd have to go to a doctor and get a strep test to find out. But like I said, it's usually fun for students to see what's growing in the throat because it always surprises them when they look and see how nasty their blood agar plates are. But that's typical and normal, right?
Strep disease or strep throat, it's very painful. Because I had one in January, it wasn't easy at all.
Yeah, if you have a strep throat, you have a sore throat, so it is painful, and you do need to get antibiotics, right? Because there are potential consequences if you wait too long, like scarlet and rheumatic fever, heart valve damage. So you always want to get a strep throat treated, with antibiotics. So anything else about blood agar? Oops. Sorry about that. But any other questions about blood? Okay, the next thing that we would have done is to try to identify the type of staphylococcus you have growing on your skin. And we're just using the traditional 3 species of Staphylococcus: aureus, epidermidis, and saprophyticus. So what you would have done is you would have taken a sterile swab, and dipped it into sterile saline, and then vigorously rubbed some portion of your skin. Not your hands, because you wash those before lab, not in your ear or up your nose, but somewhere else. And then you would inoculate an MSA plate, right, a mannitol salt agar plate, because we are trying to selectively grow Staphylococcus. And as you know, all staph grows on MSA, so all staph can tolerate the high salt. What we're looking for specifically is, can your staph ferment mannitol? Remember, if mannitol gets fermented, the pH will drop and the agar will turn yellow. So that's what we're looking for. If, after incubation, you did have yellow agar, that would suggest Staph aureus, because Staph aureus can ferment mannitol. So it's not definitive, but it does suggest Staph aureus. So most people, their MSA would not be turning yellow, but that would be the first test, is the MSA test. And then in the second lab period, you would take some bacteria growing on the MSA and inoculate a coagulase tube and a novobiocin antibiotic plate. So the 3 tests are MSA, coagulase, and novobiocin sensitivity. For coagulase, remember we mentioned that enzyme in lecture. It does form clots, and some of you may recall from AMP that we all have fibrinogen in the bloodstream, and coagulase can start cutting and cleaving the fibrinogen into fibrin, which associates to form a clot. And so in the image, the tube on the bottom has this thick clot, right? So when you turn the tube sideways, the clot is still there on the bottom. If there's no clot, well, then the liquid runs along the tube. And then again, Staph aureus is positive for a clot. It's positive for coagulase. The third test was an antibiotic, a disc diffusion test. And so you would take some bacteria from your MSA plate, dip them in sterile water just to help you spread the bacteria all over the entire surface of the agar plate, and then apply a novobiocin disc in the center. As the novobiocin diffuses through the agar, if it inhibits the bacteria, it will create a zone of inhibition. And then you have to measure. And the measurements are here on the bottom. So on the left here, we see a real small zone. So we know that's way less than 17 millimeters, right? On the right, a very big zone. If we measured it, it would be at least 22 millimeters. So the organism on the left is resistant. It grows close to the disc. On the right, the organism is susceptible because it cannot get close to the disc. So based on all three of these test results, you would try to match it up with the species of staph shown in the table. So if you had Staph aureus, your MSA plate would be turning yellow. You would have a clot in your coagulase tube, and your organism would be susceptible, right, to the novobiocin. But like I said, most students have no yellow agar, so they would use the other bacteria growing on their plate. And so they would normally see a negative coagulase result, as well as a susceptible organism. And so that would match up with Staph epidermidis. MSA negative, no clot, and susceptible. Every once in a while, we have one student, maybe two, that are MSA negative, coagulase negative, and resistant to novobiocin. So that would suggest Staph saprophyticus. But the majority of students, people in general, have Staph epidermidis on their skin. About a third of people have Staph aureus, and then just a small percentage have saprophyticus. The largest group is epidermidis, which is why the name is epidermidis, right? It's found on the skin. But about a third of people have Staph aureus, and a small percentage have saprophyticus. So you would have matched up all three of your results to see which species of staph you had, and most of you would have been epidermidis. And then the rest would have been aureus. And maybe we might have had one saprophyticus or none, right? Because often we don't get any saprophyticus. Although one semester I had two in one class. But that would be how you would determine the type of staph growing on your skin. Any questions about that? No, all good. So sometimes your results don't match. And can you think of some possible explanations as to why all three test results would not match one of these species of staph?
Maybe there was contamination of something else.
Right, contamination, because you are taking your bacteria from the MSA plate, right? So if your colonies are small, you have to use two colonies, one for coagulase, one for novobiocin. So if the colonies are not exactly identical, then you may have been testing two different organisms. Sometimes you may have a larger colony and a real tiny colony right next to it, almost touching, and you don't realize it, and you pick up two organisms at the same time. So it's possible you were testing two different organisms, which is why you got conflicting results. And what's another explanation? So another possible explanation. Let's say you have yellow agar, but it's coagulase negative and still susceptible to novobiocin. Or maybe no yellow agar, it's coagulase positive and resistant to novobiocin. What's another possible explanation as to why you get conflicting results?
Maybe you have like all of them.
Well, no, but we are testing just, one colony from MSA. So we know we've got a pure culture, but we're still getting inconsistent results. So what's a possible explanation for that? You have a pure culture, but you're still getting inconsistent or conflicting results. So you know this. So nobody has a guess? Rachel, no guess? Brian?
No, I could only think of the contamination thing.
All right, so we're not contaminated. We have just one organism, but it's still giving conflicting results. So what could be a possible explanation why?
What of if the sample, not enough sample used?
No, we've got plenty of sample, and it's pure. So why could our organism be giving conflicting results? Right, so you have an organism, it's not giving the expected results because something happened to it. What happened to that organism?
Oh, maybe it died already.
No. Something happened to it. It's alive and well, but something happened. I think you guys are thinking too hard. Or maybe not hard enough. So what could have happened to the organism, right, to cause it to give unexpected results or atypical results? change. What do we call a change?
A mutation?
Mutation, right? So you could have a mutant, right? So maybe you have a mutated Staph aureus. It can no longer ferment mannitol, but it still makes a clot. It's still susceptible to novobiosin. Or maybe it can ferment mannitol, it can make a clot, but now it's resistant to novobiosin, right? So you could have different mutations in your staph, which could give results that conflict, right? That are not typical. So that could happen.
I thought about that, but I didn't think it could happen that fast.
Oh, really? That fast? But you've had that staph growing on you, so you could have a mutated staph that's been growing on you for years, right?
Oh, right. I guess so.
Remember the video we watched, the giant agar plate, 11 days, right, for resistance, right, to occur. So it can occur very quickly. And so sometimes you do get results that don't exactly match because bacteria do have different strains, right? They have different mutations. And when it comes to doing unknowns, if you were actually doing your experiment in class, carrying out all the different tests, some of you would find that your results don't match exactly because sometimes that happens where the bacteria just don't do something that they normally do. And in fact, if you look up different bacteria and their biochemical test results, it's very common to see something like, well, maybe 83% of the time, right, that organism is positive for that test. Well, that means 13% of the time it's not, right? Or 17% of the time it's not. So it depends. Not all organisms are always positive the way that they should be. Most of the time they are, but there are strains that are not positive. So, bacteria do come in different varieties, right? They have different mutations. And so they don't always match 100%. So now when it comes to Staph aureus, remember I said about a third of people carry Staph aureus, and more of them, that staph they're carrying is MRSA. What was MRSA? What did MRSA, MRSA, stand for?
I forgot what the M is, but it's resistant Staphylococcus aureus.
Right, so the M is usually methicillin, right, that particular antibiotic, or sometimes they just say multiple resistant Staph aureus because it is resistant to many, many different antibiotics. More and more people, MRSA is becoming part of their normal flora. So they're actually carrying MRSA on their skin, even in their noses. And for them, it's now part of their normal flora. As long as it stays on the outside of the body, it's not a problem. But if it gets in right through the skin, a break in the skin, a cut, scratch, parenteral routes, it can cause a serious infection because it's so highly resistant. And so it used to be that MRSA was a nosocomial infection. Now it is becoming a community-acquired infection. So you get it from other people in the community rather than from a hospital setting. So that's very concerning, but this is what's happening now, is people are being colonized with MRSA. So any questions or comments about the skin identification, the staph identification?
So even people who don't normally take antibiotics, people normally don't take antibiotics, but even people who haven't really taken them ever can have MRSA on them?
It's possible if they got it from someone else, right, from somewhere else, but it's rare to find anyone who's never taken antibiotics these days. Almost everyone's taken antibiotics at some point. But it is community acquired, so you get it from other people. So you do need to know these 3 test results for all 3 species of staph. So this table, you need to know this. And it's actually very easy to remember because you only have to memorize 3 things. Which organism ferments mannitol?
Staphylococcus aureus.
Right? Which organism forms a clot?
Staphylococcus aureus.
And which organism is resistant to novobiocin?
Staphylococcus saprophyticus.
Right, saprophyticus. So if you know those 3 things, you know everything else in the table, right? Because everything else is the opposite. So Staph aureus is positive for MSA, it's positive for coagulase, and saprophyticus is resistant, right, to novobiocin. So just know those 3, with those 3 results, and then you know all the results in that table. So questions about staph? Okay, now we're going to talk about your teeth. So the Snyder test is for dental caries, and carie is a fancy word for what?
Cavity.
Right, for cavity. And so a dental carie is a cavity, and cavities are caused by bacteria fermenting sugars to produce acid. And then the acid eats away at the tooth enamel, and that causes a cavity. And there are 2 types of bacteria that are primarily responsible for this, Streptococcus and Lactobacillus. For Streptococcus in particular, it's the species mutans, M-U-T-A-N-S, which is primarily responsible for cavity formation in people. Both of these groups ferment sugars, glucose and sucrose in particular, and they also feed on milk protein, casein. And so the idea is that the more Streptococcus and Lactobacillus that you have in your oral cavity, the greater the risk of developing cavities. But it's just sort of a test that suggests your risk. Foods rich in simple sugars increase the risk for cavity development. So it's important to have good oral hygiene, right? Brushing your teeth twice a day, flossing at least once a day, using mouthwash at least once a day is recommended. So how many of you floss your teeth? And be honest. I know many of you don't. And the question is, why not? Why don't you floss your teeth? It really is important. So if you don't floss your teeth, here's an experiment for you to perform later. Go buy some floss and then floss your teeth. And then smell the floss. If it stinks, that's a bad sign and tells you that you need to floss, because if you floss regularly, then your floss will not stink. And the other thing is spit in the sink. If you spit blood, that tells you you have gingivitis and your gums are not healthy and you need to start flossing. So just go get some floss and do those 2 things and see, because if the floss stinks or you're spitting blood, your gums are not healthy and you need to start flossing. It's also important to brush your teeth twice a day. And then in the evening, brush, floss, and use mouthwash before you go to bed. And that will help your teeth. because it's important to do these things while you're young, because when you get older, you're going to find you need root canals, you need crowns, you need dental implants, all because you didn't take care of your teeth and gums when you were younger. So believe me, it makes a huge difference. So it's best to keep your teeth as long as you can. They've also discovered that oral health is important to your overall health. So how healthy your teeth and gums are makes a difference in terms of your overall health too. So it is important, right, to practice a good oral hygiene. So make sure you do that. And if you're not, make sure you start doing that. So what we would do in lab is each student would swab their teeth and gums. So you get a sterile swab and pretend like you were brushing your teeth. And then you would stab that swab into some Snyder agar. And then we'd incubate the tubes for 48 hours for you guys to see them. I would actually check them at 24 hours to see if anyone's tube was starting to get nasty, because normally you are supposed to check in 24 hours, 48 hours, and 72 hours. But you just get to see them at 48. But I do check them at 24, right, to see if anyone's nasty and make a note of it. So Snyder agar contains glucose, right, the sugar that's going to be fermented, casein, right, the milk protein, and a pH indicator, bromocreosol green. And the agar already has an acidic pH of around 4.8. So bacteria that produce acid, they can tolerate the acidic pH. Other bacteria can't. So that makes Snyder agar both selective and differential. Right, the acidic pH makes it selective, and glucose fermentation makes the agar differential. So the green will turn to yellow with acid. And the idea is the faster the tube turns yellow, that indicates higher levels of those two groups of bacteria, Streptococcus and Lactobacillus. So if you have a lot of those bacteria, then theoretically you have a higher risk of developing cavities. So if your tube turns yellow in 24 hours, that means you have a very high risk. If it's yellow in 48, a moderate risk. And if it just starts turning yellow or still green at 72, you have low or no risk. So here we see two Snyder agar deeps. The one on the left is very yellow, and the one on the right is still green. So ideally, you would want your tube to stay green or just slightly, start to turn yellow in 48 hours. If it's bright yellow in 24 or bright yellow in 48, right, that's not really very good. One semester there was a student, not only did their tube turn bright yellow in 24 hours, but the agar was cracked and broken. So what was happening in that tube? The agar was cracked and broken, so what was happening?
Does that indicate like CO2?
Well, gas production, right? We can't be sure if it's CO2, but it definitely was producing gas. So I've only ever seen that once, and that was not a good result, right? A bright yellow in 24 hours plus a lot of gas formation by those bacteria. So yeah, that wasn't very good. So, but the idea is the faster the tube turns yellow, the higher those numbers of bacteria are, and therefore the higher your risk of cavities. But it's just a suggestive test. Now, we don't do any controls, and we don't obviously control for the students to make sure that everyone has done the exact same thing before they swab their teeth. So there are factors that can affect these results. So what would you expect some of those factors to be that might affect your test results? Especially given that you would have inoculated your teeth, shortly after 1 o'clock.
Maybe they just
That may be food.
Right, so what did they eat right before swabbing your teeth, right? Because that could have an effect. Let's say you ate some yogurt right before you did the test. Well, yogurt has both Streptococcus and Lactobacillus in it, right? So that could skew the results. make them look much worse than what they actually are. Right? So maybe you're someone who's fastidious and you brush your teeth after every meal. So maybe you brushed your teeth right before class, right? So your teeth were nice and clean. Right, so there are possible factors that can affect these results, which is why I said it's just suggestive as to your relative risk, but it does depend on what you did right before you swabbed your teeth. Maybe you were chewing gum, sugarless gum, which does help to remove bacteria and acids from your teeth. So that could have improved, your test results. So there are other factors that can affect, right, these test results. So it's just a suggestive test, right, to give you an idea of your possible risk, right, for cavities. So questions about Snyder agar? So you've heard me talk about how you need to stop destroying your normal flora, right? Because they're protective. And yet here, with your oral cavity, I'm telling you, brush, floss, use mouthwash. You want to kill those bacteria, right? You want to reduce their numbers. Isn't that conflicting information? I'm telling you, on the one hand, for the rest of your body, antibiotics are bad, antibacterial soaps are bad. You don't want to, destroy or remove all your normal flora. But for the oral cavity, I say go for it, right? Kill as much bacteria in there as you can. Why the difference?
Maybe because they're like opportunistic for your teeth, but not in other places.
Well, most of those bacteria that thrive in your oral cavity, right, that's a very different environment from your skin and other parts of your body. So not so much, but why do you think we really need to practice good oral hygiene today, whereas 100 years ago, 500 years ago, 1,000 years ago, we didn't have toothbrushes and floss and mouthwash back then. And tooth decay was not as big a problem back then as it is now. And why do you think that is?
Is it because some of the food we eat contains are fed with some antibiotics, which may affect our teeth?
Well, not the antibiotics, but our diets have changed a lot, right? Especially relatively recently. If you look at, the human diet over the past 100 years versus beyond that time, we're eating a lot more sugar, especially simple sugars, right, than we ever used to eat. Right? We used to eat lots of protein, complex polysaccharides, not that much, simple sugar. But today we eat a lot of simple sugar, at least most people do. And so all those simple sugars are helping to create a favorable environment for bad bacteria that we don't want, right, to be growing in the oral cavity. And that's why we need to practice good oral hygiene. Unless you're going to stop eating ice cream and cookies and cake and candy and all that good stuff, you really need to brush, floss, and use mouthwash. But if you eat lots of protein and complex carbohydrates, then it's not as much of a problem, right? But most of us like sugar, right? I mean, they didn't have Doritos 100 years ago, did they? Right? So because our diets have changed, the oral flora have also changed in a bad way. And we're feeding them so much sugar, they're making so much acid, they're causing all these problems with our teeth. So that's why we need to kill them and reduce their numbers. So make sense?
Yeah.
So a surprising study was done. A researcher wanted to test some native peoples living in the Amazon. And her hypothesis was that because those native peoples, don't brush their teeth and floss and use mouthwash, she expected them to have much higher numbers and a greater diversity of microorganisms in their oral cavity compared to people that live in the city, right? Because people in the city, brush, floss, and use mouthwash. Her hypothesis was wrong. In fact, those native peoples had lower numbers of bacteria and lower diversity of bacteria compared to people who lived in the city. And it had to do with diet. In the city, we have lots of different kinds of things to eat. Out in the jungle, just a limited number of foods to eat. And so that favored a limited number of types of bacteria. But in the big city, you got all different kinds of foods, right? So you can feed all different kinds of bacteria in your oral cavity. So the researcher was very surprised because, she expected the natives to have more bacteria and a greater variety than people that lived in the city that brushed, flossed, and used mouthwash. But it was the exact opposite. So it's all the different stuff we eat. So any questions on that? So what's due on Wednesday?
Aquarium sensing.
Right, the quorum sensing, right? So make sure you guys watch that video and submit the worksheet on quorum sensing. And then for the unknowns, several of you had some problems with your unknowns, so I gave you some feedback so you can resubmit it. So make sure you go back and check that. And just in case you're concerned, we're not allowed to provide feedback unless we enter a grade. And so the grade I put is a 0 just because there needs to be a placeholder, a number there, right? So I can submit a feedback to you. But once you redo it and resubmit it, it will not be a 0, right? But make sure you go back and check your unknowns, look at the feedback. Some of you need to pay closer attention to the flowcharts and the test results, and then resubmit those as soon as possible.
Professor, in case you need some, if you say provide more information, is it just to write more?
Correct. On the description of the biochemical tests, right? What they tell you, why they tell you that.
Okay.
Right. Rachel, do you have a question?
Yeah, I did. Where can I find the worksheet for the quorum sensing?
It's under course content. Let me check and see what week. Course content week 11. That's where it says quorum sensing.
Oh, okay. So just open that document and then put that in.
Yep.
And was the video the one that you spoke about that lady speaking?
Yeah, the links are on the document.
Oh, okay. Thank you.
It's a very interesting video, but it is a little bit dated right now. It's a little bit old, so you're going to look up some of the information she talked about and see how she's wrong, or at least, based on what they knew back then.
Professor?
Oh, I love how I see Tiffany's using
Yeah, Christiane?
For the next generation antibiotics, do we have them in the market now?
Well, you have to look those up and see the things that she was talking about. Right. So what she was talking about, you have to look that up and see how that's going. then Sarah, only for those people who I gave a 0 and feedback have to resubmit. Otherwise, there's no need to resubmit. So if you got a good grade and I didn't say anything bad, then it's okay. So is there anything else?
Please, when will our results be available for the exam?
They're already done and posted. I go to the grade center.
Really?
All right, so while I was talking to you guys, I was grading them. So
Thank you.
So let's see for your class. Look up the statistics and see. Your average was 77.25. So that's pretty good overall.
So I just want to double check. The final is only if you want to drop one of your grades.
Well, the final will replace the lowest grade. So if you're happy with your grade at the end, then you're done. You don't have to take the final.
Okay.
Right, so that's just if it can help. And then you can take it. If it's higher than your lowest exam grade, it will replace that low exam grade.
So our grade is based on four tests and 2 lab tests.
And then
And
all
assignments.
the other things, right? Your unknowns, quorum sensing. So all those things together.
Got it. Thank you.
Professor, could the optional final replace one of the biological unknown assignments?
Nope, only exams. Okay, is there anything else? All right, that'll do it. So I'll see you guys on Wednesday.
Thank you, Professor.